All right, welcome to our second and final video for chapter 21. We're going to look at how viruses get into our cells and cause infection. We'll look at prions, viroids, and a little bit about the CRISPR-Cas system. So how does a virus cause disease within a cell? The damaging effects that happen within a cell during infection are known as the cytopathic effects. And this is happening when the virus is replicating and leaving the host cell. So these include lysis, and I saw this earlier in the previous video when the viral particles burst out of the host cell to spread. In some cases, we saw that lysis does not happen and rather the virus buds out of the cell, leaving it alive to continually shed viral particles. In some cases, apoptosis or programmed cell death of the host cell will occur. And sometimes just the immune response causes disease, or the symptoms and signs of disease as the immune system responds to that foreign particle, the virus in the body. Basic steps of viral infection include these four shown here. The first is attachment, and this is why viruses can be species-specific and also tissue or cell-specific. This is because viruses have to bind to specific receptors on the surface of the host cell before they can enter the cell. And the receptors will bind to those glycoproteins or spike proteins that are either present on the surface of the capsid protein shell or on the viral envelope. So viruses can only attach to cells with the correct receptor to bind those glycoproteins, those spike proteins. Then the virus enters, which usually means that the DNA or RNA enters the cell. They replicate themselves and assemble the viral pieces until they form those virions, which will eventually lyse out of the cell or bud out of the cell. So here's what entry looks like, and I wanted to show you this picture because ultimately the virus has to get its nucleic acid into the cell, and it can inject the nucleic acid, or here I can see this is a virus with a protein capsid shell. The nucleic acid has to get in. If it has an envelope, it has to shed its envelope, open up the capsid until the nucleic acid is released. In every instance, you're trying to get the nucleic acid into the cell. If for some reason you cannot open up the capsid protein or open up the envelope, then the virus will be non-infectious. It won't be able to cause disease if the nucleic acid cannot be released. Here's an example of influenza virus infection. So this is an envelope virus, as I mentioned earlier. It enters the cell when the spike proteins on the envelope bind to the correct receptors of the target cell. Here, it enters through endocytosis and then releases the viral contents. So it releases the RNA into the cytoplasm, which then enters the nucleus so that we can replicate the RNA and use it to transcribe the viral proteins that are needed to assemble and produce new virions, new viral particles that will eventually bud out of the cell. In this slide, we have a different envelope virus. This one is HIV that we saw earlier, also enveloped and also an RNA virus. It uses its spike proteins to bind to specific receptors on the surface of certain cells in our blood to get in. Once it gets in, you can see that it releases its RNA, and it uses its reverse transcriptase enzyme to synthesize DNA. Once the DNA is made, it enters the nucleus, and it integrates into our own DNA, into our own chromosomes, and it's hiding there. In that stage, we call it a provirus. While it's hiding there, it can be latent, so this can be an infection where the patient does not have any symptoms yet, but secretly the virus might be creating messenger RNA, creating viral proteins, and starting to bud out of the cell. Just looking at the impact of different viral infections on humans, you can see you probably have heard of many of these viruses and their subsequent infections. One common example are sexually transmitted diseases that are caused by viruses. STIs, or sexually transmitted infections, Generally, if they are bacterial, we can usually take antibiotics to treat it and cure the disease. But if it's viral, you pretty much have it forever for the rest of your life. It's not true for all of these viruses that we see in this picture, but for the STIs or STDs, this is true. Because many of these viral infections are so severe they can result in death of many patients who are infected, we have luckily created vaccines to prevent infections from many of these viruses. So vaccines, their purpose is to warn your immune system of the virus so that in case you ever encounter it in any daily situation, your immune system is prepped and ready to fight the virus. There are three major categories of vaccines. Some of them are attenuated vaccines, and this means that you're getting an injection, or this is usually not an injection, you're getting exposed to a weakened form of a live virus. Because the virus, it's not live because we said they're not made of cells, but the virus is still infectious, so there's a small risk that patients who receive this type of vaccine can get the disease, the viral disease, although a less severe form of it. Attenuated live virus vaccines are, you might have heard of the flu vaccine. That is a spray that they spray in your, like kind of your nose or in your mouth. This is usually the attenuated flu vaccine, which has a weakened version of the flu virus, and it could give you the flu, although it will be less severe and usually a shorter duration of the sickness. But the benefit to this, so the drawback is you might get sick. The benefit is that you have, or this type of vaccine offers the strongest type of immune response. So your immune system will be able to quickly fight the infection if you encounter it again in the future. Another benefit to this is that if someone gets the weakened version of the virus and gets sick from that, that weakened version of the virus can spread if someone gets someone else sick next to them. A bunch of people get sick from the weakened version of the flu virus. That spreads the vaccine, but also allows the protection to spread as well. Another type of vaccine is the completely killed virus. Here you cannot get sick from that virus. The virus is no longer infectious, but your body is still able to recognize the virus and attack it in the future to offer that immune response. The benefit of this is you don't get sick, but the drawback is your immune response is not as strong. Finally, the safest probably type of vaccine is a subunit of the virus. So if this was the virus, you're only getting a piece, an antigen that is injected into your body or into your blood. So your immune system will recognize that part of the virus in the future so that if you encounter the virus in the future, you can attack it. You're protected. Besides vaccines, other ways that we can target the treatment of viral infections is through different drugs that directly block viral infection at different stages of infection. So earlier I showed you a picture of how HIV infects the host cell. So these are the different types of drugs that we have these days. We have fusion inhibitors that block the virus from binding to the host cell receptor in the first place. We have reverse transcriptase inhibitors that prevent the viral RNA from converting to DNA, so it can't integrate into our chromosome. We have integrase inhibitors that block integrase enzyme, and that's the enzyme that HIV uses to integrate its DNA into our genome. We have protease inhibitors, which prevent the maturation of the virus, so it cannot infect future host cells. So there are many drugs that we have these days to treat HIV, for instance. The problem with RNA viruses, as I mentioned earlier, is that RNA viruses mutate pretty often. And what happens is with each cycle, with each host cell cycle where they're replicating and budding out of the cell, you might have mutations in the RNA that have created alternative versions of the spike proteins. So now your fusion inhibitors no longer work, or there are mutations in other components of infection or other steps of infection. So now your drug no longer works and you have to find a new drug to target that specific mutation. So this is constantly happening in our fight against HIV. We're constantly developing new drugs to attack HIV with those new mutations. And we see something similar for the virus that causes COVID-19 as well. So the virus that causes COVID-19 is known as SARS-CoV-2, and SARS-CoV-2 stands for Severe Acute Respiratory Syndrome Coronavirus 2, and COVID-19 stands for Coronavirus Disease of 2019. So this happens to be an enveloped virus that is an RNA virus. So similar to HIV and influenza, it gets into the cell, releases its RNA, and during replication of the virus, as I mentioned, RNA viruses have a pretty high mutation rate. So as they mutate with every life cycle, what often happens is that these mutations lead to changes in their spike proteins. So perhaps the vaccine that we got 2 years ago to attack a certain spike protein on the previous form of the virus no longer works because of these mutations. Now, this virus has a new spike protein, so we have to make a new vaccine for that new spike proteins. And it's a constant race for the virus to evolve and adapt so they can survive, or not really survive because they're not alive, but so they can continue infecting host cells, and then for us to continually fight off these new mutations as well. So now we're going to switch gears, and we're not talking about viruses anymore. We're going to talk about another non-living infectious particle, and these are known as prions. Prions are proteinaceous infectious particles. So these are proteins that are infectious and cause disease. These are really interesting because they're extremely small, misfolded proteins that cannot be destroyed through cooking your food. So they can be found if humans eat meat from animals that have prions. There are no nucleic acids in prions. It's just protein, just an infectious protein. But the disease that they cause are severe. So these cause severe, fatal neurodegenerative diseases that result in death, including mad cow disease, variant Creutzfeldt-Jakob disease, Kuru, scrapie, and more. So as I mentioned, we cannot destroy them by cooking. So sometimes people say, oh, I can just make sure my hamburger is well done and then I won't have to worry about it. That's not true in this case because these prions will still be infectious. The only way we can destroy prions is really through complete incineration, like burning something until it's completely charred. Recently, we have new techniques, and they've created something known as prionzyme that might be able to inactivate prions, but it's not really out yet. It's not really well known yet. What's scary about these prion proteins, some people call them prion proteins. I've heard both. I think both are fine. Is that abnormal prion protein, if it touches normal prion protein that's found in our brain, our nervous system, then it converts the normal prion protein into abnormal prion protein. So it's almost like playing tag, where the bad version will convert the good version to the bad version. Tag your it, for instance. And this results in degeneration of the nervous tissue. The signs and symptoms of infection include loss of brain tissue, dementia, which is a loss of memory, judgment, behavior, language, thinking. Individuals will lose their muscle coordination over time, and you can diagnose this with a brain biopsy, but unfortunately there's no treatment. There's no cure, and really you just have to do as much as you can to alleviate the symptoms and provide really ultimately end-of-life care. Finally, we have our third infectious particle, which are viroids. Remember, we talked about viruses, which are also known as virions. We talked about prions, which are infectious proteins, and our third category is here, viroids. Be careful not to mix them with viruses. Viroids are infectious particles that are only made of RNA. So they're circular RNA molecules. And so far right now, we only know that they infect plants, whereas prions we saw earlier can infect humans and other animals. Viroids infect plants, and these can self-replicate. The RNA molecules can self-replicate. They don't encode proteins, but what they do when they infect plants is that they bind to messenger RNA of plants and they inhibit protein expression. They inhibit translation so the plants can no longer make their normal proteins. This results in crop failures because these proteins are usually involved in growth of the plant. So I have normal potatoes here, and these potatoes have been infected by viroids, which means that they cannot make their normal proteins, and that results in stunted growth. Here's another picture. These potatoes have been infected with viroids, and here's a normal potato. I don't know, I feel like all the potatoes I buy from the store look like this, so maybe they're all infected by viroids. I don't remember ever seeing a potato that looks that great. The last topic for this video is the CRISPR-Cas9 system, and it's actually not in chapter 21, but I wanted to talk about it because it's important in terms of biotechnology. It's a relatively new technique that we've discovered. We discovered it in bacterial cells, and it's related to viruses because it's a means of bacteria to protect themselves against viral infection. So really interestingly, this is a picture of a prokaryotic cell. This looks like something like E. coli. And bacteria like E. coli have the ability to remember previous infections from viruses. Every time they're infected by a bacteriophage, which is a virus that infects bacteria, they take a piece of that viral DNA and insert it into their CRISPR DNA region as a spacer. So these black diamonds are CRISPR DNA, and the colored squares are DNA from different viruses, from previous viral infections. What happens is if the same virus that has previously infected them attacks again, they go back to their database of previous infections, find the matching phage DNA, recruit an enzyme known as the Cas9 enzyme, CRISPR-associated protein number 9, and they will look for the complementary sequence on that phage DNA and cut it. Here it is, here's the cut. They will cut it so that the phage is inactivated. The phage or viral DNA is inactivated so it cannot replicate in the cell or create new viral particles. And CRISPR stands for clustered, regularly interspaced, short palindromic repeats. These are the black diamonds that are here, and again, they're palindromes that are regularly spaced. So why do we care about CRISPR-Cas9? The reason is previously, before we discovered this technique, we were limited to where we could cut DNA. We primarily could cut DNA at restriction sites that were recognized by our restriction enzymes or restriction endonucleases. If you had a gene of interest you were trying to cut out, but it did not have restriction sites, then you basically couldn't work with that gene. Now that we have CRISPR-Cas9, we can cut DNA anywhere we want by manipulating the sequence that will be recruited or will be recognized by the CRISPR-Cas9 system to cut the DNA. So scientists can switch out the viral DNA in the spacer sequence with the DNA sequence of interest. That will allow CRISPR-Cas9 to cut at any sequence of DNA you want. All right, that takes us to the end of chapter 21. Thank you so much for listening.